So we had seen in the very first lecture on biodiversity various maps on the distribution of species richness. Yeah. So biogeography is the field which basically looks into the aspect how these spatial patterns of biodiversity and the distribution of species on the planet are controlled. Yeah, what are the mechanisms? So if you look at such a map, for instance, species richness, as you've just seen, or just the occurrence of specific species or of plant or animal communities, we realize that they vary spatially in non-random, so more like, I wouldn't say regular, but in a non-random way. Yeah, often along certain gradients, climatic gradients or spatial gradients. Yeah. So this is one observation we can say. So that seems to be that there are certain forces which result in these patterns. We see certain patterns in the distribution of biodiversity. And if we look at different regions on the planet, yeah, for instance, let's say you go to Central Africa and you go to Brazil. If a helicopter drops you there, you think, okay, that looks pretty much the same, you know, so you're in a rainforest. But if you identify the species present, you will realize, okay, they are very different. Yeah, the trees will be different and the animals will be also largely different. So that is, even though we have maybe similar climates and similar environmental conditions, soils, nutrients, etc., different regions have different distinct assemblages of species. Yeah. So the biogeography, we try to understand this spatial diversity. Yeah. So we try to understand spatial patterns of biodiversity across space. And also this can be done, again, as we had heard before, what biodiversity encompasses. It can be studied on the scale of genes all the way to the scale of ecosystems, yeah, or biomes as it is called. And biogeography consists of maybe two main approaches. One field is called historical biogeography, which is largely concerned with identifying the historic processes, mechanisms, etc., which result in the modern spatial distribution of biota. Yeah. So historic means here evolutionary processes, geological processes, paleoclimatological aspects, paleoecological aspects. Yeah. So historical biogeography has a strong temporal perspective on things. In contrast, there's ecological biogeography, which looks more like how in terms of distribution. Yeah. So it's very strongly focused on the interaction of species. Yeah. So it goes into questions of competition, but also niches and all kinds of things. Yeah. There's a field which is called phylogeography, which is really rapidly growing over the past 20 years or so, which is concerned mostly with the spatial patterns of genetic signals or structures. Yeah, so you can measure the genetic composition of one species, or you compare the genetic makeup of multiple species, and then look into these aspects. And then if you're a paleontologist and you study extinct organisms, you can also do paleobiogeography. That is, you look into the distribution of species in the past, let's say dinosaurs, for instance, or brachiopods or whatever. Yeah. So these are the main ways this field is made up. So I mentioned historical biogeography. So today we start with aspects, how can we reconstruct the development of lineages, biotas. Yeah. And then later on today, if we make it, we talk a little bit about the classification of geographic areas or regions on the planet based on their assemblages of species. Yeah. So these two things are major themes in biogeography. If you look more closely, we try to explain differences in the number and types of species between geographic areas and also along certain gradients. Yeah. This and the next point, explaining variation within or between individuals or populations of closely related species, is more what we are going to be concerned with when we read these case studies, these papers, and discuss them. Yeah. So we have a planet which hosts a tremendous amount of species or other levels of biodiversity. Yeah. So let's think about how we've heard about how species originate in the last class, yeah, on Monday. So we know there are all these modes of speciation. We know that selection is relevant and so on. But how do we study actually how these organisms formed and how their distribution is influenced? So we have to get familiar with the major tool of phylogenetic trees. Yeah. So this is just this is so essential today in ecological evolutionary biogeographic studies that we cannot ignore this approach. So we have also seen a phylogenetic tree last time when we talked about speciation, and I show the same tree here. You remember that species today are normally classified according to monophyly, so they go back to a common ancestor. And the clade then is a group of monophyletic related organisms. Yeah. One thing you have to be, or you have to realize is that every phylogenetic tree is only a hypothesis. This is not like a fact, which is like written in stone. So these trees can look very differently depending on how you build them. And there are other reasons why they are not perfect. Yeah. But let's first explain how a tree, or what does these trees show us. Yeah. So here on this figure, you see a phylogenetic tree. You see that it consists of branches. So these are the horizontal lines. And then where these lines split, you have a node. Yeah. And these nodes denote a common ancestor, typically a most recent common ancestor. Yeah. You have terminal nodes. So these are the tips. And the tips are represented mostly, let's say, by species today. It could also be a higher taxonomic level, let's say a genus, a family, an order, whatever. It could also go below the species level. But let's assume this is species. And here we have like these four plant species, those four flowers. Yeah. So they, according to this phylogenetic tree, form a monophyletic group, a clade. Yeah. There's an outgroup. So outgroups are more distantly related taxa, and they allow us to root a phylogeny, so to really see the relationships. Is there anything else? Oh yeah, and the length of these branches, so the length of the horizontal lines, such as this one or this one here. I hope you see my mouse. This length is proportional to the evolutionary divergence of these organisms. So often it reflects time. But in order to have it on an absolute time scale, we need to use either a molecular clock, we need to calibrate the tree by fossil data or by other geological ages. I come to this in a second. Yeah. So these trees are relatively simple hypotheses or models because they are based on a number of assumptions. one assumption is that two species go back to a single common ancestor. So divergence is typically into two descendant species. Divergence is according to how these trees are drawn, instantaneous. And instantaneously, you would assume also reproductive isolation. These things are evolutionary, not realistic. But they are reasonable on large evolutionary timescales, and in particular when uncertainties in time are considered at these nodes. So for instance, the assumption that that all things go back to a common ancestor is violated in the case of eukaryotes. So all organisms with a nucleus go back, this is the hypothesis, by the symbiosis of archaea and bacteria. So you have two, so this is kind of where this assumption would be violated. But all organisms would go back monophyletically to the last universal common ancestor. This is what genetic data predict. So the question is, why does it need to be two descendants? Couldn't there be multiple descendants emerging at the same time? So the assumption is just that we talk here about like cladogenesis, so the splitting or the formation of new species. And of course, this is not, again, an assumption which may be true, but biologically it is reasonable to assume when you have certain mechanisms that first you have two populations which slowly divide from each other before they become reproductively isolated. So having more than this requires more assumptions. And often these phylogenetic trees are based on the concept of, what is it called, parsimony. So you produce trees with the least number of assumptions. But maybe sometimes, or perhaps you can see where three branches descend from one node. That is possible, I think, but it's rare. And I must say, I'm not producing phylogenetic trees because I'm not working with such data sets normally. So if you really want to know the details, you need to talk to the people in the biology department who teach this stuff, like Ralf Tiedemann and maybe the ancient DNA people around Michael Hofreiter. But let's continue here. So how do we reveal that organisms are related? So before we can draw a phylogenetic tree, we have to understand certain aspects of evolution. And one thing which is essential, is that related organisms share similarities that are derived from common ancestors. And these characters or traits we call homologous. So, or a homology as a noun. So before we can produce these trees, we have to look into, we have to compare. Let's say you have a collection of animals or plants in front of you. You compare characters, certain traits, whether it's whether you extract the DNA and compare sequences of DNA, or you look at the morphology of a flower or the color pattern on the wings of birds or something like this. You would sort them according to which look more familiar. But the thing is, you have to identify characters which were shared by common ancestors. So if you just look at the presence of DNA, which is a homologous character for all life, but it goes back all the way to the last common ancestor of all life forms today, that doesn't help you to build a tree. So this field, phylogenetic systematic, really was advanced in the 1950s or 60s, I think, by the publication of one work which defined rules how to classify so that you can build correct trees. And this goes back to a German entomologist, Willy Hennig. Yeah. And this classification of organisms is based on shared, derived, or novel characters, which Hennig called synapomorphies. Yeah. Relationships on trees are never based on the absence of characters. Yeah. And even though species evolve at different evolutionary rates, at some time they will accumulate differences, yeah, which are not shared by other species. Yeah, so these are unique characters. Let's look how this looks on a tree. Yeah, so by using these weird terms from Hennig, which have made it into the international literature and which were very influential in systematics. We have here phylogenetic trees. Here we see a character which is unique to one species. Yeah. And we can use characters, let's say the chin. The chin is an autapomorphy for Homo sapiens. If you compare the chin with like great apes, they won't have like, it's not extended like with us. So this is an autapomorphy which distinguishes us from other species. But in order, that doesn't help us if we want to know with whom we are closely related. So what we are looking at are synapomorphies. So these are shared characters which were novel at some point. Yeah. So if we identify those characters, and let's say with great apes, if you look at chimpanzees, maybe we look at brain size or other things. Yeah. So you have to identify those characters. How do we do this? Yeah. So here you see a so-called character matrix. So on the one side, we have a list of species, yeah, and then we list a number of characters, yeah, presence, absence in this case. And these characters are synapomorphies, so they are not present in all. Yeah. And the taxa with the largest number of similar characters are then more closely related. So with this character matrix, we can build a tree. Yeah. So we have here the character 4 legs, amnioid eggs. So for those who don't know what this is, these are, so an amnioid egg is an egg which has like a skin, which protects the embryo from drying out. Amphibians don't have this, for instance, fish don't have this, yeah. In German, with humans, it would be the Fruchtblase. I don't know what this is in English. I think it's also the amnioid sac. In any case, there's hair, there are mammary glands, and there's live birth. So we have these characters, and then we can build a tree by looking at the presence of these synapomorphies, yeah. So we look, does the fish have legs? No. So it splits off. Then 4 legs is the synapomorphy for all these other species. Yeah. The frogs don't have amnioid eggs, so they fall out. Crocodiles, platypus, kangaroos, and elephants have those, so they are one clade. Crocodiles don't have hair and don't have mammary glands, so they fall out as next. So this way we can build a phylogenetic tree based on certain characters. Yeah, this is a very simple example. We can add 2 more characters, a pouch and a placenta. Yeah. And these are then autapomorphies for elephant and kangaroos. So we can split those groups, yeah, based on these characters which are not shared. Yeah. So kangaroos could be like placental mammals and, marsupials, and elephants could be placental mammals. So this is the very basic origin of how these trees were built, yeah, prior to the development of molecular techniques. You must imagine, however, that you never do this with single, if you do a species phylogeny, you don't use single characters. You use a whole number of characters. Yeah. So you measure a lot of things, which was done classically in morphology, measure the length of certain bones, the width of bones, etc. And then your matrices become much bigger. Yeah, you compare many more characters, and that's where it really becomes difficult, because this can result in various solutions to a phylogenetic tree. And today there are lots of statistical methods to produce them. Yeah, there's certain yeah, there are Bayesian statistics. There are models based on what is it? Distance measures on a tree, and then there's what is called maximum likelihood. So this should not concern us here, but just remember, we should always compare things in terms of the presence of characters. Yeah. So we can use this concept also when we do molecular phylogenetics. Yeah. Instead of, let's say, using coloration, anatomy, or other things, we use other data sources on the molecular level: protein structure, amino acid sequences, genome structure or genome composition, genome length, DNA sequences. We can use mitochondrial DNA, nuclear DNA, chloroplast DNA with plants. We can measure the abundance of RNA or RNA sequences. Yeah. So obviously the best is to use DNA. Originally, before DNA sequencing was used, mostly molecular phylogenetics was done on the proteins. But remember, the last time you've seen that different amino acid can build the same proteins. So, the best is to use DNA sequences. Yeah. So we also heard that mutations change sequence of DNA. So over time, the DNA of two divergent converge. Yeah. So if we want to find homologous characters, so sequences which were shared, we look and compare sequence data and see which ones are shared and which are different. So so we can put up similar character matrix. Here you have a list of species again, beetles, and on the top we have 38 characters. Each site here for a nucleotide is character state, and then we can map out. And here you just color code these things based on the presence of cytosine, guanine, thymine, or adenine. Yeah. And then you can sort these things into a tree. Yeah. We can use amino acid sequences, we can use nucleotide sequences, and this is an example for mammals. And you can derive trees based on both data sources. You can also look how consistent these are. Yeah. So let's take an example. Here we have the sequence of one gene for chimps, orangutans, humans, and rhesus monkeys. Yeah. And in the middle, you see in black highlighted the nucleotides which differ. Yeah. So can you build, or could you propose a tree based on these sequences? Yeah. Thinking about what we've just talked, what are the rules are, similarities, and shared characters. Yeah. Can you propose? Yeah, if you have ideas how the tree can look like, or which organisms or which of these 4 species are more closely related, just type it in the chat. Yeah, I like to hear like at least 2 answers.
So I will say rhesus monkey is more related to chimpanzee, and then orangutan and humans.
Okay, so why do you say chimps and
I mean, I will say that rhesus monkey is the one that differ the most from all the 3 because it has an extra
yeah,
sequence at the end, the A and G, if I'm correct. I don't see very well, it's too small.
right.
So, and then, yeah, and then it looks like the orangutan. Well, I think I'm also guessing because I read one time that orangutans are more related to humans than other species. But I also see with the coding of the DNA, it seems like they differ very little.
Mm.
Yeah, but
Yes.
I guess if you look closely at the chimpanzee and the human, we've got like the big black bars that where like all 4 are differing. You see that they don't have the same point mutation that they have between orangutan and chimpanzee, for example. So they're still the same. So they're really just differing in just one nucleotide, and this is the single mutations that you've got both at the beginning of both strands. See what I mean? So like in chimpanzee, the first mutation is the C, followed by the T, followed by the C, followed by the A, by the TG. But the second mutation of the chimpanzee is not a mutation in the human, as well as the 4th and the 5th and the 6th. So essentially they're just differing by 3 mutations, right? Mm-hmm. Whereas orangutan and rhesus monkey got the same point mutations, where, yeah, so I'd argue in a different way.
Okay, yeah, that was good. Yeah, I think we have to refer here, as we just heard, to this last a group here. We see these ones, TG and TG, make chimps and humans more similar, right? And the CA and the CA here put orangutans and rhesus monkeys more closer. Yeah, and this is also seen here and seen there. So these 3 character sites, as we call them, are decisive. Yeah. This is just a simple example, but you see, it is not always a straightforward thing. If you have more species, you will have to do more careful examination of these sequences. And as I said, there are various tools, statistic tools, and software programs which help you producing these trees. Yeah. Indeed, chimps and humans are most closely related. Yeah, so we diverged around 6 million years, and I think what I read is that there was gene flow perhaps between both groups. I wouldn't call it Homo sapiens, but Homo, or until about, or not Homo, but the human lineage until about 4 million years. Yeah. Okay, so the good thing is if you use nucleotide sequences, we have some advantages in comparison to morphology. Yeah, so it's less subjective which characters we choose. We have only 4 letters on a sequence which we can compare. So it's a very clear way of comparing things. Perhaps less prone to selection and convergent evolution. I show you an example in a minute what this means. And we have large data sets. That's the advantage. Yeah. So here is a morphological phylogeny for the major order of mammals. This is the one I've learned when I was a student. Yeah. So, and I highlighted one group here, which are the insectivora, the hedgehogs, shrews, etc. And you also see how certain groups are related to each other. Yeah. If I plot the molecular phylogeny in comparison, then we see a large rearrangement on the relationship between these mammalian orders. And we see the loss of the insectivora. Yeah. So the insectivora were shown to be paraphyletic. Yeah. So they were not a monophyletic group. They included distantly related families in genera. What the molecular phylogenies on this high order taxonomy showed that there's a clade which is termed Afrotheria. And this shows, and these two groups, the Afrosoricida and the Macroscelidia, so these are elephant shrews and tenrecs, so very interesting creatures in Africa. They were part of the insectivora, but are not related. Instead, they are more closely related to elephants and Sirena, which are the manatees and those guys. This came out by using the molecular phylogeny. So today, if you do your species, if you want to describe the relationship in a family or genus of species, it's always good to combine both approaches. Yeah, so morphology has not been abandoned, still really relevant, but it shows that molecular data provide us often very new insights, which morphology wouldn't predict. And one of the reasons, what I mentioned before, is that morphology is prone to what we call convergent evolution. Convergent evolution means that under similar selection regimes, very similar adaptations evolve. Yeah. And in organisms which are not closely related. Yeah. And this here shows these Afrotheria, and they compare this to this group of Laurasia-theria. So these are animals which evolved in Laurasia, which is the continental mass, which was once the connected landmass of North America and Eurasia. Yeah. And we see that molds are represented by these golden molds in Afrotheria. Hedgehogs are by these tenrecs, which live in Madagascar. Then we have shrews, and we have similar shrew-like animals there. We have the dolphins, and then we have the dugong, and we have the pangolins and the aardvark. So these animals look morphologically similar. If you only have the bones in front of you, you may classify them as the same, but they don't show close relationship based on the molecular data. So what an morphologist thought was the same, perhaps in particular with these groups up here, which have been split, is the result of convergent evolution. So we can now use these molecular phylogenies to actually date the timing of the divergence of two species, or more than that. Because over time of divergence, the nucleotide sequences or amino acid sequences will also change, and often in a linear manner, because mutations accumulate with time. So it's similar, this idea is that these substitutions change at a constant rate has been proposed to be an analog to the ticks of a clock. And you can also, if you're a geologist, think that it's similar to the decay of radioactive elements. So this is the assumption of the molecular clock, that the substitutions occur at constant rates through time. And if I know at which rate this occurs, I can calculate the age of a divergence of two taxa, for instance, as shown in this very simple example. We have a common ancestor with a certain nucleotide sequence. This common ancestor splits into two organisms, or two clades, or two species. And the substitution for these 10 nucleotides is 1 per 25 million years. If I know this rate, I can calculate the timing of the last common ancestor, the timing of the splitting or the divergence. This is the assumption. However, it has been shown that molecular clocks, or strict molecular clocks, are somewhat too simplistic, because rates of molecular evolution can vary between organisms. Can you just, I think we can hear you, if you just say a few reasons why this may be the case.
Well, I guess if you've got, for example, a duplication event happening, then this does not, like a duplication of a certain gene or sequence, then this does not mean that this is followed by a functional difference, which would lead to two different genes being activated. And
okay.
So I guess you can have different mutational events happening at different rates, which end up in different phenotypic expression.
Okay. Any other ideas?
It's more of a question, but would the rate, the frequency of these ticks of the clock, would they be different depending on the length of the genome?
That is possible. That's a good question, and I must say I can't give you a conclusive answer here, but I can indicate it. Let's say, because it has shown, this is also not. This is more like a general observation. It's not like always the case that organisms which evolved later in history have, or what we sometimes call more complex organisms, have longer genomes. So the question is then, does a longer genome mean that there is a higher likelihood for mutations to occur than in a smaller one? So it is a question of like having basically material available for mutations, and the more you have, the more mutations you will actually can produce. But there's an important point, that the reason why substitution rates may be different is shown that one thing is just simply think about the generation times of organisms. If an organism reproduces 4 times a year, so it builds gametes 4 times a year, it has more chances to accumulate mutations than if it reproduces only after 10 or 20 years of its existence. So, and there's actually, it has been shown that there's a negative relationship between the body size and the substitution rate, because another reason is why mutation rates differ between organisms is metabolism. So there are a number of reasons why this assumption of a fixed clock may not be correct. And what people do then is that they either use an average value and vary it, or they let the rate evolve over time. it is important that when you apply a certain substitution rate, that you try to use it if a rate is kind of like empirical determined somehow, that you use it from an organism which is most closely related to your system you study. Yeah. So using a rate which is produced, let's say, for mice, yeah, and you apply it to a certain group of plants, that might not be the correct approach. Yeah. And we will probably read papers where we think, what kind of substitution rate have these guys used? And then we see, hey, they used it from a completely different group. So this assumption then might be different because it has been shown that the substitution rates differ between species. Yeah. So this, of course, makes this whole thing complicated. But we can solve it somehow by using fossils. If we use fossils, we can calibrate the timing with an absolute timescales for these nodes. Yeah, so these divergence events. Yeah. So because the fossil record is incomplete, and it is very unlikely that we always deal with the oldest fossil of a species, yeah, that these fossil calibration points provide us with minimum ages. Yeah. So let's say a fossil is dated to 20 million years. We can use the 20 million years as a very fixed calibration, but it's more scientifically sound to give that age a range. And often this range is being pushed towards an older age. So our 20 million year old fossil calibration point may actually be then extended to 30 million years. Yeah. And here I show you a calibrated tree. I think we might read this paper, I'm not so sure. And these stars denote the calibration points. What often happens, and which is surprisingly not the case in this study, is that the fossil calibration points occur earlier than the divergence or the nodes in the phylogenetic trees based on molecular data. One reason is because I just said that the fossils are often not representing the oldest possible age of a split, but often also because these substitution rates predict older divergence. Yeah. So you solve this issue by having these uncertainty ranges here, which are these blue bars. So you can shift this divergence event, this node, over, let's say here in this case, almost 5 million years. Yeah. So another way to calibrate a phylogenetic tree based on molecular data is, in certain cases, you can use geological dates. Here we have a group of birds, the honey creepers, which are endemic to the islands of Hawaii. Because Hawaii are volcanic islands, we can date the earliest timing when these islands had formed. So when volcanism was basically above the water. Yeah, and there's this method which we will talk about, argon dating. Later we'll hear about this method. But these islands have different ages. Yeah, so some islands are over 5 million years old, others 4, some are around 2 million years, and Hawaii, the main island, is just 400,000 years old. Yeah. With that, we can calibrate our phylogenetic tree. So the endemic or indigenous honey creepers of Hawaii should not be older than the age of the islands. Yeah. But do you have any ideas what could be wrong with this approach, especially with these gray bars? Why could this provide wrong calibration points in time? Just throw out something.
Because birds can migrate.
Good point. So they can migrate. Does it mean that the divergences are too early or too late? So exactly, so there's the other answer we got, that we don't know when they came to the new islands. And that's exactly what people were arguing. with this approach, you can imagine that one island, if it formed 5 million years ago, provides 5 million years of time for evolution. So you can have dispersal events which postdate the origin of the other islands. Yeah. So not all individual taxa, which perhaps are now endemic to the other islands and don't occur on this one, for instance, but only on Hawaii main island, like these two guys here. Yeah. They could have also originated there and then dispersed over there and died out over here. Yeah. So this has been contested in the literature, and but it shows you that this is a very interesting case where we can use geological data to look at least some constraints on the timing of this radiation. And this brings me now, unless you have further questions on phylogenetic trees, we go now more into some aspects of diversification. Yeah. So we see here these honey creepers. And I have another question to you, and I want to hear at least 3 answers, because this is a simple question. We had last time, we talked about geographic modes of speciation. Which mode of speciation do you assume for this group of birds? Okay, allopatric. So we have allopatric. Any other ideas or suggestions? So we have allopatric, peripatric, sympatric, and parapatric. So we have 4 possibilities. We have heard allopatric. So anyone who chooses sympatric or something else? Parapatric. Parapatric, okay. Peripatric, okay, super. So we heard peripatric twice, but it seems nobody is up for sympatric. We can do this the whole afternoon, but we don't have the time. I tell you, it's funny. This is, okay, the first event. Yeah, so we have an outgroup. This is the common rosefinch. But the most common ancestor, this event here was peripatric. Remember that a group has to split off from its main range and colonizes a new place. It's a form of allopatric speciation, but it's called peripatric. Yeah. So for this event, when this species here, which invaded these islands initially, diverged so much that it was a different species from its mainland South American source, then this was a process of peripatric speciation. We had a founder effect, if you remember this. Yeah. But this whole radiation is largely sympatric. Yeah. Allopatric would mean that barriers had formed. But there, of course, there might be barriers between these islands in terms of some ocean, but they could fly across these places. And think about that most islands have actually a large number of birds which occur on these individual islands. So they developed in sympatry or evolved in sympatry. Yeah. And this is a special case of diversification, which we will talk about. It's called adaptive radiation. Yeah. So if you remember, going back to our first lecture, we've seen the theory of island biogeography. We know that on an island, we have an immigration rate and extinction rate, producing an equilibrium number of species. And otherwise, we also have to think about speciation. Yeah. So what is an adaptive radiation? When 2 species split, yeah, because they share certain characters, they are very similar. And they are not just similar, but also in their ecology. So they probably feed on similar things, or they reproduce in a similar fashion. They perhaps evolve in similar habitats, etc. Yeah. So if a monophyletic group diverges or evolves into distinct species which occupy a broad range of ecological niches, then we have an adaptive radiation. Yeah. So think about 2 species have overlapping niches. So they exploit the same resources. Then they cannot coexist. One will outcompete the other. Now, this is the concept of competitive exclusion. They can only coexist if they segregate their resource requirements, if they somehow feed, for instance, as a bird, on slightly different resources. Yeah, this is the realized niche. Yeah, so they compete each other. They might eat more or different forms of food, but if they co-occur, they have to split so that they are not going extinct, or one of them at least. So that means that if we have co-occurrence of closely related species, they tend to differ in their resource use. Yeah? I'm not giving you an example now. We just look at these Darwin finches. Yeah? So these niche differences can be morphological, can be physiological, behavioral. If they did occur, we call it character replacement. Yeah? And this is seen here in these different builds of these Darwin finches. We heard about directional selection or disruptive selection the last time. And these things will happen if we have enough resources available. So on Hawaii, on Galapagos and other places, we can have one founding effect, founder event, where some bird gets there. And then if these islands are new, there's no competition with other species, these populations of this founding individual or this founding population can start to exploit various food sources or resources and thereby slowly diverge from each other. Yeah. This is what we call an adaptive radiation, and it's a specific case for diversification. Yeah. So we see monophyletic groups being present in certain areas. We have sympatric speciation. The other example I mentioned the last time are the cichlids, fishes in East African lakes. Yeah. So adaptive radiation occurs when competition exerts strong selective pressures, and then divergence happens when ecological opportunities are present. Yeah. So can you give me examples under which situations this could occur, except for the invasion of an island? I want 3 answers.
I suppose if the niche is very small or is becoming very small, then the species that are present would have to start separating their resources differently.
Mm-hmm. Can you specify why the niche would, get more narrow?
Well, for example, maybe with habitat fragmentation, if there's a forest that's being fragmented by roads that are being developed or something like that.
Okay.
I'm not sure, I think we already had an example like last week or something about the Darwin finches that were adapting to a drought. So their beaks got smaller so they could feed on the smaller seeds instead of competing with the other finches who had bigger beaks and were like competing for the big seeds that were still available.
Good answer. So it's really, the point is that we need different niches. Yeah, they have to be, so the first of all, we need a common ancestor, which has a broad niche. Yeah, and then we can basically exploit different available niches by specifying on specific parts. Yeah, and this can happen obviously on areas which are free of competitors. Yeah, and then this one species can split off. But it also occurs often after mass extinction events because dominant groups just disappear. And it can also happen when you evolve a new trait, yeah, a new organ, or somehow you're able to exploit a previously inaccessible area, for instance. And this is, for instance, the evolution of wings. Yeah? And I show you a few examples by means of phylogenetic trees. So here are the Galapagos finches. They are in adaptive radiation going back to one common ancestor. and they have evolved under intra-specific competition into this group, which possesses all kinds of different beaks. They all exploit different food resources, different niches. Yeah. And they are not divided by mountain ranges or other things. They can fly from one island to the other. Yeah. Or they could like be moved by wind in some cases. Yeah. Here's an example of a presumed adaptive radiation for all or for most mammal groups after what was traditionally called the KT boundary, the Cretaceous, which is now the Paleogene boundary, 65 million years ago. So that's when this bolide asteroid impacted the Earth and led to the mass extinction of dinosaurs. With that extinction, you open a large number of niches where the dinosaurs were feeding on. And so with that, you have a diversification in herbivores and etc. in various groups. Yeah. It's still contested because phylogenetic trees also give different ages. Some say that all these groups originated already before the extinction of dinosaurs. But this is a possible way or a possible theory.
I have a question.
And then here I show you phylogenetic trees for early groups of birds. With the evolution of wings, a new territory, which was previously unexploited, and where like
Sorry, did you hear us?
insects were available to catch, where you could escape predators, etc. This was available and led to a very fast radiation in birds at evolutionary rates faster than contemporaneous dinosaurs. Yeah. So this happened also during the Cretaceous. Okay, so this was an example, adaptive radiation, if we talk about islands or if we talk about certain specific events in Earth history. This is something. Okay, there was a question from Charlotte.
Yeah.
Yes, so the question is, could mammals emerge and diversify after the extinction of dinosaurs, because dinosaurs preyed on mammals? Sure, this is also true. Yeah, so the size of mammals was relatively small during the Mesozoic, even though recent paleontological finds have shown that at least beaver-sized mammals were around. Yeah, so they were not just like a shrew or a mouse, but obviously large herbivores were, for instance, absent. So in the predation by dinosaurs was certainly a reason why these mammals maintained smaller body masses to be able to escape. Okay, I hope this answers the question. So, okay, so these adaptive radiations are a sign of diversification in certain times or in certain places. Yeah, and this brings us to the topic of biogeographic patterns. Yeah. So we talked a lot about time now, and now we are going to look into space. Yeah. By the way, here's a book by Alfred Russel Wallace, who was essentially the father of zoo geography. So this is the first book on topic over 100 years ago. If we look at our Earth, we can have different scales of observation. We can look at the entire globe, we can look at continents, we can look at regions, populations or individuals. Yeah. And remember, we can look at alpha diversity, beta diversity, or gamma diversity, yeah, for example. If we look at biodiversity distribution on the planet, we can realize we can measure certain aspects of spatial patterns along, for instance, gradients of latitudes, gradients of longitudes, gradients of elevation, for instance. We can compare island biotas with that of continents. Yeah. So there's various perspectives we can do. First thing we want to look at are distributional ranges. So where and how are organisms distributed on the planet? We have some major types. We have restricted ranges, which we call endemic. We have separated, spatially separated ranges, which we call disjunct. and we have widespread ranges, which can be cosmopolitan. Ranges are dynamic, they change over time due to climatic, environmental changes, competition, evolution. And then we can finally, and that is what Wallace did in his book, we can divide the Earth into certain provinces or realms, which show that in these areas, specific assemblages of species co-occur. So let's look at these types of ranges first. If a species is restricted to a specific geographic region and occurs nowhere else, we call it an endemic. Of course, this term is a little bit ambiguous because all organisms are endemic to Earth. All African species which live in Africa are endemic to Africa. So we have to specify this. But the opposite of an endemic, usually as it is, if we look at endemic, not at everything which is endemic to Earth, but endemic to specific regions, the opposite is a cosmopolitan species, worldwide distribution. Can anyone give me an example of a worldwide distributed species? Okay, so what I see here is mostly, so I see rats, ants, pigeon, mice, flies, humans. Humans is a good example. Rats obviously is the result of human expansion as well. Flies, this is not, so we want to look at species, so fly is a whole family of, or an order of insects, Diptera. So this is two. Mice is also a large group of, so of course these taxonomic groups can have global distribution, although, and pigeons, I think as a bird group, they may do, I'm not, okay, that's good. I give you one species, the fern bracken. It's in German Adlerfarn. Pteridium aquilinum is the Latin name. I'm not sure if it's globally distributed, but I've seen it in North America. It occurs here in Brandenburg, on Berlin. It's a very common fern. It occurs in Africa. It occurs in Asia, Southeast Asia. I'm not sure if it occurs in Australia or in South America, but it's very likely. One reason why these ferns often have, can have global distributions is because they produce spores, and spores can easily be transported across the planet. In any case, the last type, disjunct distributions, are populations which are found in geographically separated regions. And here we have the marsupials, in German Beuteltiere. I don't know the French name of it, or marsupial. And they are found only in Australia and South America. So obviously very strongly separated. So for endemism, we can distinguish narrow endemism and broad endemism, depending on how large the actual areas are. So narrow endemism would be, for instance, a very localized occurring species. Let's say you have one species only occurring in one lake. There are very many cases where individual lakes harbor only one certain species of fish. This is a local or narrow endemic. Broad endemics would then occur, let's say, for instance, all species confined to Central Europe, for instance. And then there's a distinction between neo-endemism, so recently evolved endemism, so that an organism which evolved, let's say this fish which only lives in one lake, may hadn't had the opportunity to expand its range. Paleoendemism is when formerly widespread species, like ginkgo, for instance, the tree was formerly much more wider distributed in the Mesozoic than it is now, where it only occurs in a few places in China, where it is an endemic, essentially, before people started to plant it outside. So this is an example of a paleoendemic. So I give you a number of maps just to really cram you in that these different range types. So the lemurs are an example. These are primates. They are endemic to Madagascar, the whole superfamily of lemurs. Yeah. But the mongoose lemur is endemic to northwest Madagascar. Yeah. The Iberian lynx, distinct species of lynx, is endemic to the Iberian Peninsula. It does not occur elsewhere, yeah. Although it is declining in its range, yeah, it's facing extinction, although it's doing a little bit better now. The fossil record doesn't show that it occurred outside the Iberian Peninsula in the past. Yeah. So disjunct distributions consist of two or more separated groups. Yeah. It simply means that the range is discontinuous, and it can result from either range expansion or contraction. So expansion would be that population or individuals migrate off and found a new population elsewhere, founder effect. Yeah. So they cross a barrier, for instance, and then they start to form. Then you have a disjunct range. It can also occur when the range contracts and gets separated by, for instance, the formation of a mountain range within a range of species. Yeah. Can also happen if we destroy habitat by cutting a forest apart. Yeah. So here, this is an example of an anthropogenically fragmented range. So this is the present disjunct range of the wild African dog. Yeah. It was formerly a continuous range. Humans have driven it into distinct fragmented range patterns. This is an example if we look at the two species of gorilla we've mentioned before. The western gorilla occurs here, west of the Congo River, and the eastern gorilla on the other side. So this is unclear whether there was founder effect producing this species, the species migrating north of these rivers and then down here, or whether there was once a continuous range which split by some type of environmental change. Here we see this butterfly, the Apollo butterfly, and its distribution in Europe is restricted to the Alps, to the highlands of the Pyrenees in the Balkan Peninsula, the Ural Mountains, and it occurs in the lowlands of Scandinavia. This is a sign of what we call a boreal-alpine distribution, and these are relict occurrences after the Ice Age. During the Ice Age, the species was much wider distributed because cold climate and the food plants occurred more widely. And with the warming during the Holocene, during the past 10,000 years, it was restricted to these relict habitats. Yeah. Here I show you a couple of disjunct patterns for plants. Yeah. This orange is Fagus, this is beech. Yeah. So we have two beech species in Europe, as far as I know, yeah. And then there are beeches in Japan and East China, and then in Eastern North America. They are disjunct. The genus has a disjunct distribution. Every continent has different species, however. Same with the genus Notophagus. Yeah, the southern beeches. They occur in South America, Australia, New Zealand, and New Guinea. We can have disjunctions which are outside the tropics. For instance, these crayfish. Yeah. They occur on every continent, essentially, outside the tropics. But for instance, here we see that this has a disjunct, this species or this group has a disjunct distribution in North America and East Asia. Here's an example where we have the magpie, Elster in German, a bird which occurs across Eurasia. And then we have the azure-winged one, which occurs in East Asia and curiously only in the Iberian Peninsula. So what is a major theme of biogeography is to explain these patterns. Now, is this a result of range contraction or range expansion? Is it the result of dispersal or of vicariance? Yeah, has the range been split by some kind of separation by a physical barrier? Yeah. Here we can look at another bird. This is the red kite, yeah, Rotmilan, and the black kite. The red kite is endemic to Central Europe, or to Europe at least. Yeah. So Germany has a high responsibility for the species, but it has a continuous range, more or less. The black kite is not cosmopolitan, but it occurs on a much larger range, continuously. Yeah. Palms are a group of plants which have pantropical distribution. So they are not cosmopolitan, but they occur only in the tropics. This is also an interesting pattern. If we look at monkeys, we would see the same for this family. So I ask you, give me an example for a cosmopolitan, oh, we had this, okay. So a cosmopolitan group of animals are the bats. This particular group of bats here shows cosmopolitan distribution. And you can tell me why this is so, right? That answer is not correct. So second answer is correct. So because they can fly. It's very simple. If you have means to overcome barriers, you can invade many places. Yeah. So just being able to fly gives you an advantage compared to somebody who has to walk. Yeah. Okay, so we know that ranges are changing over time. Yeah. So if you look at these changes in ranges, we have to consider different timescales. Yeah. So here I give you maps for the distribution of oak, yeah, a tree which is typical for Europe, or tree species, the genus Quercus. And you see here maps for the distribution of oak over the past 12,000 years. On the left and on the right lower hand side, you see it for the British Isles since about 10,000 years. And these lines, these lines connect points. So how do we do this? We go and collect sediment samples from lakes or bogs and determine the pollen. We date those sediments, and with that we have a record when a species of plant started to occur in an area. If we combine then a large number of sites with pollen records, we can draw what this is called basically it's a contour line. It's a line of similar percentages or the similar timing of the occurrence of pollen in a certain region, by combining sites. So these isopole maps give us the range expansion of oak after the Ice Age. Yeah. It's a very good tool to look at range changes for plants. Yeah. We can also monitor this. So for instance, we can look at how starlings, European starlings, which have invaded North America after they've been released there in New York, how fast they spread across the continent. So we can measure those rates and say something how quickly these things happen. We see also the opposite in terms of human intervention. Bison had been restricted from a formerly continuous range into like two very small separated ranges. Yeah, these pink dots. So this can happen over historic times when humans are often the cause. We can also model future changes in the ranges, for instance, by modeling the distribution of species. We know under which conditions they occur, and then we can say, okay, what happens if we double CO2, which is something which unfortunately is probably occurring. And then we know how the climate is changing, and we can look how these species respond and predict their future ranges. And here's the case of this beech, Fagus grandifolia, the North American beech, and we see that its range, which is here, is probably then be restricted to the Appalachian Mountains when that had happened. So we have range contraction. So why do species change their ranges? And this has something to do with dispersal, the way they move. And we have two ways. We can actively move This, however, requires that the organism spends energy. Then it can fly, as we've just seen with bats. It can dig, walk, or swim. Flight, as is mentioned here, allows for long distance dispersal, so you can overcome large areas. We know that certain migrating birds fly across almost the whole world every year. So this is possible. We can also have vagrants. These are, for instance, exotic birds of North America, which sometimes are swept over the North Atlantic by winds. So this is rare events. Then we have passive dispersal, where just things are being moved by other agents. So for instance, over long time scales, continental drift will move organisms from one place to another. Just with the moving mass. But wind can, for instance, disperse like little insects or spiders. You can also hitchhike on another animal. For instance, a tick on a bird can fly from one continent to another. And plants use so-called diaspores, fruits, seeds, or spores, or pollen to disperse. So then we have different types of dispersal. Jump dispersal, which is a long distance dispersal. So this is often facilitated by storms. Then there is diffusion. So the gradual expansion, as we have just seen with these oaks, of a range of a species over several generations. So it took oaks 5,000, 6,000 years to reach north of the Alps, from their ranges during the Ice Age. And then we have secular migration. This is a migration which takes so long that over time we have actual evolutionary change of the organism. So it is 1 minute before the lecture stops. I think we should stop then. Sure. Yeah. Okay. Are there any more questions? We will continue here next time. And I give you a home assignment to read one paper on the aspect which we've just discussed, dispersal and disjunct ranges. And I want from everybody next Monday, I hope that the lecture will not last the entire time, but that we have about 20 minutes to discuss these papers. And in order to facilitate the discussion, I like to have 2 questions on the paper from each student posted on the paper before. And these questions should be, or could be twofold. They could either, you want to know something, how the researchers did it. You can ask a question, or we can discuss things where you propose an alternative solution to what researchers propose in terms of how they explain the dispersal scenarios. Is that clear? So I don't want the question, where was the researcher born who wrote the paper? This is not of interest to us. So we want to know either you have a problem with understanding something, or...
Can you repeat the first question?
Yeah.
Well,
so,
it's 2 good questions.
So 2 good...
No stupid questions.
Yeah. So the first question is, just if you need clarification with something you haven't understood, then ask a question. For instance, how did the researcher do X, Y, Z method? Or something like this, So a question on the method is fine. So if you really want to know how did they do this, or let's say, The other question you can propose or is like, could that not also be in this way? So question the results and perhaps propose an alternative, or just question how consistent this is, or something like this. Is that okay? And one last question to today's lecture. Was this okay? Was it too much? Or did you learn something new? Or was it just all stuff you already knew? Because we have students with different backgrounds, and sometimes I'm struggling with like, how do I put my lecture together here? And I want to get some feedback to ensure I'm not presenting to things which are so well known that you're bored, but also I don't want to put too much complicated things in here. Compiling these lectures and changing them from last year, it's a lot of work to ensure that it makes sense, but we want that before we discuss papers that everybody understands these concepts. Yeah. So I'm happy to hear that this is helpful to you and new. Okay, then I wish you a good rest of the week. Stay safe, stay healthy, and have a hopefully good weekend.